The first talk in the next session is from Jia Nee, and she's going to talk about SMPD1 in Parkinson's disease.
Thank you very much, Peter, for the kind introduction. And I would like to thank the organizers, especially Jia, for inviting me here to share my work today. So it took me about more than a day, about a day of traveling to get here, but it's so worth it because, it's so nice to meet all of you finally in person. And many of you have been, I've known many of you as names on papers for years, and it's nice to come and hear about all the great work going on GBA1. So let me start off this session on other players in the pathway by focusing on one of them, which is SMPD1 and its role in Parkinson's disease, which I will refer to as PD. Okay, so the confirmation of GBA1 as a Parkinson's disease gene has actually raised interest in a lot of the other lysosomal storage disease genes. So like GBA1, most of them actually encode enzymes important in lipid metabolism. And we know that deficiency for these enzymes cause autosomal recessive diseases. Often they are severe with onset in infancy or childhood. But perhaps like GBA1, they may also, a partial enzyme deficiency for these other enzymes may also Oh, this one.
It's not streaming well.
Okay, so so perhaps like GBA1, partial deficiency for these enzymes may also lead to increased PD risk later in life. And of particular interest is of course SMPD1. So SMPD1 encodes for acid sphingomyelinase. It's a phosphodiesterase that catalyzes the conversion of sphingomyelin to ceramide and phosphocholine. And as many people have pointed out, it does share a common product with GCase, which is the ceramide. And homozygous loss of function or homozygous mutation in this gene is known to cause Niemann-Pick disease type A or B. So this landmark paper by Jia and colleagues back in 2013 actually generated a lot of interest in the field of PD genetics. So in this study, I think they focused on founder LSG gene mutations identified in the Ashkenazi Jewish population. And out of all the variants they looked at, they found one L302P in SMPD1 associated with increased risk of Parkinson's disease. But this variant seems to be absent or very rare in most other populations. Since then, many other groups have tried to replicate these findings, and we were one of them. So back in 2013, we only sequenced 99 PD cases and 99 controls by whole exome sequencing. We did take the variants found only in cases and tried to genotype them on exome chip in a larger sample size. So far, we definitely did not see L302P in our samples, but we did find another variant that seems to be associated. But then again, this variant was extremely rare and very difficult to validate, the R591C variant in SMPD1. So it's been more than 10 years since the initial report of SMPD1, and many studies have been published, with some showing support for association and some reporting no support for association. So why was I invited to present this in 2024? So let me backtrack a bit to introduce the work that I'm doing in my lab. So I'm a geneticist. I'm no expert on lipid metabolism or lysosomal enzymes. I'm a geneticist focused on gene discovery, and I tend to go wherever my gene discoveries take me. So like many of you, we do the GWAS studies to look at common variants with more effects, and of course, whole exome sequencing also to look for those that are rare, but possibly with larger effect sizes. And because we don't have easy access to large numbers of large families, we tend to do it on the population level on sporadic cases. And one major gap in genetic studies has been raised a few times earlier today is really the lack of diversity of samples being analyzed. So for those of you who are not so familiar with this graph, it's simply showing the number of samples of each ancestry being analyzed by GWAS over the years, compared against the global population distribution on the right. And Asian population, both East and South combined, make up more than half of the world's population. But you can see they are still severely underrepresented in genetic studies. So I believe there's a lot more to be found from studying other populations, not just Asian, but also the African, Latino, and other lesser studied ancestries. So this was one of the motivation behind us forming the Asian Parkinson's Disease Genetic Consortium. It is led by E.K. Tan, who many of you probably already know. And all the work I'm presenting today is done on samples collected by this consortium. They currently come from at least 6 different regions in East Asia, and we look forward to expanding the sample collection more with our new collaborators and also with the GP2. So we did a whole exome sequencing discovery study on close to 10,000 exomes, consisting of about 4,300 PD cases, 5,500 controls. This work is led by Elaine Chiu, who is here at the meeting, and she's presenting her poster tomorrow. So we look forward to further discussion with all of you. So there are many ways of analyzing whole exome sequencing data, but this is the main approach that we took. So out of all the variants that we discovered by whole exome sequencing, we focus on those that are non-synonymous, predicted pathogenic. We use polyphen 2 and lofty. We filtered those that are rare, less than 1%. Above 1% is typically covered by GWAS studies. And then we did stratified GMH test as a gene-based test across more than 17,000 autosomal genes. So the idea is simple. So we are simply looking across the genome to find out which genes are actually enriched for what we classify as rare pathogenic variants in PD cases compared to controls. So from this analysis, we have about 3 genes coming up as exome-wide significant. The top hit is not surprisingly GBA1, and I think the most common variant driving it is probably the L444P variant. Rank 2 is HLA, is a HLA gene that showed protective association, but this finding was somewhat difficult to validate because of the complexity of this region. And to our surprise, what came up second in risk association actually SMPD1. But I think we were also a bit apprehensive at that time because we know this gene has been extensively studied over many years and hasn't been truly confirmed as a Parkinson's disease gene. So given that so many studies have already been done on SMPD1, it was important for us to go back to the previous studies and see if we could actually validate our findings and to see if there are any differences in what they found with ours. So we actually focused on the three largest studies that have analyzed more than 500 Parkinson's disease cases. They did equal resequencing of matched controls. And there were 3 studies that we focused on, 1 of Chinese ancestry and 2 of European ancestry. So they also provide the full variant list with carrier count in cases and controls. But because every study used a different criteria to filter for rare pathogenic variants, so we re-annotated and re-filtered the variant list to standardize with our discovery study. So this gave us a replication data set of over 5,500 cases and 5,600 controls. So from a combined analysis of the discovery and replication cohorts together, we observed quite a strong association across nearly all the cohorts that we analyzed. But we were still not fully convinced because still not sure why it wasn't fully confirmed before. And does functional prediction algorithm really matter? So could it be because we all use different methods for functional prediction? So we actually wanted to try and develop an unbiased in vitro assay to look at the effect of each variant in one standardized setting. So actually we took nearly 2 years of work to generate about 126 cell lines. So there were 125 variants that we found from the discovery and replication cohorts combined. And we generate cell lines we use a HEK293T cell line because it doesn't express endogenous SMPD1 as confirmed with Western blot. And we overexpress wild type and each SMPD1 variant at similar levels. So each SMPD1 variant was expressed at similar levels compared to wild type, with the exception of some like this frame shift mutation shown here. So we also confirmed that the overexpressed enzyme is being localized to the lysosome using the lysotracker dye. So we did functional assays. We took the cell lysate and standardized the protein amount input into the assay. So we use a substrate, HMU-PC substrate that's from a published method, and it gives a fluorescent product that we can measure to indicate enzyme activity. So each assay was done in triplicates, And the person running the assay is actually blind to case control status of each variant. And we gave each variant a functional score based on percent activity compared to the wild type SMPD1. So, okay, I mean, how do we interpret the functional score? So first of all, we try to see if they are reproducible and consistent in independent repeats of the experiment, and it seems to be they show similar results in repeats. By polyphen prediction, those predicted deleterious, not surprisingly, have lower functional scores than those that are predicted benign. By protein domain, those in the catalytic domain and the saprocin domain seem to have lower functional activity. And also looking at Niemann-Pick disease patients, out of the 125 we tested, 44 have been previously reported in patients, in the autosomal recessive patients, and out of them, 18 have been classified and ClinVar as pathogenic or likely pathogenic. And out of these 18, close to 80% had less than 50% activity measured with our assay, which I think supports that the functionally deficient set of variants seem to be enriched for true pathogenic disease variants. So we tested 125 variants that we found and saw a whole spectrum of effects. So some showed little or no effect on enzyme activity, some reduced enzyme activity to 10% or less of the wild type, and this includes L302P. And they are colored by the polyphen prediction. So those colored in blue are predicted deleterious, those colored in orange are predicted benign. And you can see there's some correlation, but it's not perfect. And I think the misclassification may actually greatly affect our interpretation of the results. So using this spectrum, Elaine actually modeled all possible thresholds to see which threshold can best discriminate a Parkinson's patient from controls. And we came up with these results that people who carry a variant that reduces activity to below 44% seem to be associated with a more than twofold increased risk of Parkinson's disease. So this is, of course, an average reading because each variant may have slightly different effects, but this is the average that we got across all the variants. And the p-value is a lot more significant than what we got with functional prediction alone. And note that this effect size is actually a lot more smaller than GBA1. So this could be why it has been a lot more difficult to confirm SMPD1 association, as it's a lot more sensitive to functional misclassification. So we also look at age of onset in the SMPD1 and GBA1 carriers. So consistent with previous reports, our GBA1 carriers also had an early age of onset and faster progression compared to non-carriers or SMPD1 carriers. So SMPD1 carriers, although the age of onset was slightly lower across all the cohorts, it's not statistically significant, so we are unsure whether it actually is truly different from the non-carriers. Yeah, this is actually consistent with also the smaller risk effect size we saw for SMPD1 compared to GBA1. And although we did gene-based association tests, we note that our association was actually largely driven by one lead variant, which is P332R, which has about 30% activity in our assay. I think people who carry this variant make up about 80% of the stat that we classified as functionally deficient carriers. And this variant alone actually reached close to genome-wide significance when we include a replication cohort. So looking at all the other SMPD1 pathogenic variants combined together, we do also see a similar odds ratio, suggesting that this effect is not limited to this P332R variant alone. So likely other functionally deficient variants also play a role in the PD association. Based on the largest whole exome sequencing study in European ancestry cohort that is led by Cornelis and with the GP2 consortium, they did not find SMPD1 at all, and we think that the differences between our findings could be likely attributed to population-specific rare variants. So they saw association at LOC2 driven by the G2019S variant, but we did not because this variant is not present in Asians. And also our lead variant P332R in SMPD1 is actually absent in European ancestry samples. So this really highlights how rare variants tend to be population or ancestry specific. And I think by studying more diverse ancestry groups, you may likely find new rare variants and genes, even in well-studied diseases like Parkinson's disease. So we then did a bit of further work to try and tease apart the roles of the two genes, like how different or similar they are. So looking at expression in the GTAC database, both genes seem to be expressed across multiple tissues, not just in the brain, and I think this is already known. We also look at human midbrain single nuclear RNA sequencing data sets, where Elaine integrated data from 4 published studies. And both genes are actually expressed across nearly all cell types. So although you see some, they are expressed across all cell types, and at rather low levels and a rather small percentage of cells. So although you may see some larger dots here, these are also not more than 10% of cells expressing it. So we just want to confirm at the protein level as well. So we did some IHC and Western blot in postmortem brain tissue, and somehow the distribution of the protein of both proteins seem to be rather uniform across the tissue. So it seems to be expressed across multiple cell types. And we also tried to address the question of whether the two genes interact, and I think so far it's been quite difficult to do so. So people who carry variants in both genes are extremely rare, not surprisingly, because these are all rare variants. In our discovery data set, we only know 8 individuals carry protein altering variants in both genes. 2 patients carried known pathogenic variants, so of course the L444P in GBA1 and the P332R and SMPD1. But their clinical features seem to be quite unremarkable, and the age of onset was variable, so nothing much that we could conclude at this time. Based on the single nuclear RNA sequencing data set, we did not really see co-expression of the two genes in the same cells, and that's similar across all cell types. So that's suggesting that the two genes are not likely to be co-regulated. And we think that further work is needed to understand their relative effects on perhaps levels of different ceramide species in different cell types. So I think further work is still needed there. So now that we have confirmed association of a second lysosomal storage disease gene, what about the rest? So based on our existing data, we did not see strong evidence for association of the other genes, but I think we are not able to rule them out without larger studies in more diverse ancestries and also more functional work. So from sequencing 10,000 samples in our discovery data set, we had only 2 to 3 genes reaching axonal significance. So looking at statistical power, we had sufficient power to detect a large effect like GBA1, but although it's very rare, low combined carrier frequency in the population. We could also detect a smaller effect in like SMPD1, but at a larger combined carrier frequency because of the P332R variant, collectively in more than 1% of the population. And we think this is really just the tip of the iceberg, because I think there are still some variants, still genes with variants with similar effects as SMPD1 or GBA1, but are present at lower combined carrier frequencies that we are still not able to detect in a whole exome study. So indeed, larger samples will be needed for further work. So I hope I've convinced you that rare functionally deficient variant, SMPD1 variants seem to be associated with increased risk of Parkinson's disease. And this association was replicated in samples of Asian and European descent. And we find that classifying variants with functional assays can help to improve gene-based association test results, at least in the case of SMPD1. And analyzing diverse ancestry group, I think, will help in our discovery of novel rare variants and genes associated with disease. And finally, impaired sphingolipid or ceramide metabolism is likely involved in Parkinson's disease pathogenesis, and we look forward to what further studies will show for this. Yeah, so I will end off by thanking my lab member who contributed to this work. Most of the work was done by Elaine, supported by Moses and Michelle, and many collaborators who have supported and contributed to this work. So thank you for listening.
That was a great presentation, and it's going to be a lot of questions in lunch, but I'm going to try to move carefully. One thing I wanted to point out, though, is that the amount of ceramide generated in the lysosome from sphingomyelin is at least tenfold greater than the amount generated from glucosylceramide, maybe closer to 50-fold greater. So if you're thinking about traffic control, if you think, sort of the ideas that I presented were interesting, this is gonna be important too, because they're both involved in generation of ceramide. So, anyway, we're gonna forego questions for now, okay?